So I'm going to go ahead and get started. We are going to talk about aspects of MHC Class II today. And then when we are finished, I will give back exams and talk about exams. Because I don't want you to be just like looking at the exam while I'm talking today. So that's why I have to do it at the end. So last time we talked through the steps of processing in the MHC Class I pathway. If you remember, we had a series of 5 steps that we saw. So the first step was the acquisition of antigen. Then we had proteolysis. Then there is delivery. There is binding and display. For class 1, the acquisition of antigen was infection. So this cell didn't really have to do anything too fancy. For proteolysis, we had ubiquitin and the proteasome. We talked about how delivery was really the sort of crucial step for class 1, and that delivery was done by tap 1 and tap 2. We then talked about binding, which was sort of facilitated by the chaperone tapicin. And we saw the process of display, which was constitutive in the biosynthetic secretory pathway. So this was Friday. Today we're going to go through the same process for MHC Class II. Recall that MHC Class II has 2 chains, the alpha chain and the beta chain, that come together to make the peptide binding cleft. One of the things to be aware of, I told you that you aren't able to get proper folding of class 1 unless you have the heavy chain, peptide, and beta-2-M. That is also true for class 2. You really only get proper folding when you have both the alpha and beta chains as well as a peptide in the cleft. But one big difference is that with class 1, we have this beta-2-M protein that is non-covalently associated. So it sort of has an on and off rate. With class 2, the structure that we're going to make is extremely stable. It is in fact stable in SDS. It's known as an SDS stable complex. And so the extreme stability of this complex is going to be an important thing that we're going to have to see today. MHC Class II binds to the T cell receptor along with CD4 in order to present to CD4 positive T cells. And unlike MHC Class I, we don't have a ton of cells doing this process. So with class 1, remember, it was all nucleated cells. We're doing that presentation process. For class 2, this process is only done by B cells, macrophages, and dendritic cells, and not by other cell types. And so this is going to be a bit more limited. In fact, these cells can sort of turn up or down their amounts of class 2. It's not as constitutive as we saw before. So this is the figure from your textbook that shows both the class 1 and class 2 pathways. Again, we are going to see later on one place where I'm going to edit this figure because there's one thing about it that really bugs me. With class 1, we had what are sometimes called endogenous antigens. Basically, they're proteins that are coming from the cytoplasm. They're proteins that are somehow already in the cytoplasm, whether that's infection, whether that's a self-peptide in the cytoplasm. For class 2, we are going to see what are known as exogenous antigens being presented. These are antigens that come into the cell by phagocytosis. And so all of the peptides that are presented in class 2 are coming from some part of the endocytic pathway. And so again, we talked a little bit last time about this idea of which side of the membrane is stuff on. Is it on the cytoplasm side or away from the cytoplasm? Class 1 was presenting the cytoplasm stuff. Class 2 is going to be presenting the not cytoplasm stuff, stuff that's in one of these types of vesicles. So the cell didn't really have to do anything to acquire antigen for class 1. The cell just got infected. For class 2, things are a little bit different. The cell actually does need to actively do something in order to acquire antigen. The easiest way to think about that acquisition is that we can think about phagocytosis. And so our cell will have either some type of pattern recognition receptor that can bind to the microbe, maybe some type of Fc receptor or complement receptor, all of which may lead to phagocytosis. We're going to envelop that microbe within the membrane and make a compartment called the phagosome, and eventually that phagosome will fuse with the lysosome. Then we will call it the phagolysosome, and we'll be killing that microbe. And so we can talk about acquisition for class 2 as being phagocytosis. In sort of basic cell biology terms, phagocytosis is just one of many ways that things can be internalized into a cell. I'm showing this figure on the left because what I want you to realize is that phagocytosis involves the internalization of something pretty big as far as the cell is concerned. You can see how large this particle is that's being phagocytosed. There are other types of internalization that are much smaller. You can also see that for phagocytosis, that cell needs to reorganize its plasma membrane a lot in order to take in something that big. It actually is going to be reorganizing its actin cytoskeleton a lot in order to do this. So this is a very active process, unlike what we saw in class 1 where the cell was really passive. Here the cell is really working to acquire the antigen. But as is also shown here, this is not the only way that things can be internalized. And so you can see that materials can be internalized in cells into smaller types of vesicles. And some of those smaller types of vesicles can also be involved in MHC Class II presentation. And so phagocytosis is sort of the famous way we can acquire antigen. But many of these, so officially clathrin-coated vesicle, non-coated vesicle, and calveoli, all would be types of endocytosis. And so we can also have some of these smaller types of internalization of endocytosis where we have an antigen brought in. We will still make some type of smaller vesicle, and it will end up looking pretty similar. So this could be phagocytosis. It could be endocytosis. And I'm going to be saying one other thing about endocytosis momentarily. As you can see, I'm leaving some other room above endocytosis because I'm going to write something else there, even though I'm not that tall. Yeah, Mark. So macropinocytosis is usually taking in relatively small things that are, and usually it's more of a fluid-based system. You tend not to get the same kind of vesicles at the end. So we can see at the top the sort of fate of material that gets phagocytosed. We do phagocytosis. We're enveloping this big thing. It goes into a phagosome because it came made by phagocytosis. And eventually that phagosome will fuse with the lysosome. It turns out if we internalize something by endocytosis, we make a smaller vesicle, but it ends up being about the same process. Here we call the compartment an endosome because we brought it in by endocytosis. Sometimes we'll see the endosomes that will have some changes happen to them. They might be early endosomes or late endosomes. And eventually we'll get fusion with the lysosome. It's the exact same process. And so we can see either of these bringing in material for MHC class II. If we look back at the type of cells that can do MHC Class II presentation, there are three major types of cells that can do MHC Class II presentation. What are those three cell types? Yeah?
B cells, macrophages, and dendritic.
So B cells, macrophages, dendritic cells. B cells are phagocytic cells. Sorry, macrophages are phagocytic cells. Easy. Dendritic cells are phagocytic cells. Easy. B cells are not phagocytic cells. And so things are a little tricky with B cells. It turns out that B cells can bind antigen using their B cell receptor, and that can induce endocytosis of the receptor as well as the antigen that is bound to it. This process is officially called receptor-mediated endocytosis, because the thing binds a receptor, and that makes it do endocytosis. So B cells are able to acquire antigen not through phagocytosis. Instead, they bind antigen with their B cell receptor and perform receptor-mediated endocytosis to bring that antigen in. Eventually, the receptor will get recycled back onto the surface of the cell while the antigen will get degraded. Yep?
Is there a specific immunoglobulin constitution that's getting?
No, because remember, this is the B cell receptor. This is not a secreted antibody. And so all of the B cell receptors are easily internalized. So we also have to have a short further discussion of cell biology to think about the names of some of the compartments we're going to be seeing today. So again, for today we're largely going to be in this biosynthetic secretory pathway. So in the places that are shown in yellow on this cell on the left. We may be in the vesicles that are going out, like the secretory vesicles, or the Golgi. We may be on the vesicles coming in, like the endocytic vesicles going to the lysosome. In reality, all those vesicles are sort of connected and talking to each other. And you can also see that here, that when things come in, they will come into an early endosome. They may eventually go to a late endosome, either by movement or that early endosome might itself mature. We may then see fusion with the lysosome. Maybe there's an endolysosome. In this process, these compartments are getting increasingly acidic. So the lysosome is super acidic. The early endosome is a teensy bit acidic. And we have sort of increases in that process, or increases in acidification throughout all of these steps. There is one other type of compartment that is sometimes found in this set of related compartments, and it is known as a multivesicular body, or an MVB. The exact details of MVBs are slightly confusing, and there's a lot that we don't totally understand about MVBs, but we can see situations where the endosome can sort of endocytose itself and make little compartments within itself. So you can see what was originally on the outside of the early endosome sort of becomes a compartment within the compartment. It has multiple vesicles, so it's multivesicular. It's a multivesicular body. This, we think, is really important for the degradation of transmembrane proteins, because how else would they get degraded if they were still on the outside? Here, they can totally get degraded because they're on the inside. This seems to be a compartment that is found sort of between endosomes and lysosomes, maybe around the late endosome. They are pretty easy to see under a microscope. I will mention the multivesicular body a little bit in this presentation process, and so I wanted to make sure you knew what the multivesicular body was. So at this point, we have acquired our antigen for MHC Class II. We now need to do proteolysis. Happily, we have acquired our antigen into this system of vesicles that lead to a place where we do proteolysis. So the lysosome, where we sort of end up this pathway, is actually a compartment where we can do proteolysis. So as we see those materials moving from early endosome to late endosome to lysosome, they're going to more and more acidic places. And we are seeing more and more proteases that are able to degrade those molecules. And so you can see that the lysosome is super acidic, and it's full of nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases. It's full of all the enzymes, as well as being very acidic. And so just by the nature of getting to the lysosome, our antigen is going to get degraded. Most of these are acid-activated enzymes. So you don't want to destroy your whole cell. So you have enzymes that only work in acidic conditions in the lysosome. And you also hold them in the lysosome so they're not destroying the rest of the cell. But we're able to use the lysosome, which is basically the trash compartment for the endocytic pathway, to degrade our antigen in the case of MHC Class II. Yep, Lexi.
Is providing the hydrogens that activate the enzymes?
So the hydrogen pump is what's making the lysosome acidic,
Okay.
which is,
So
yeah.
the enzymes act all the time?
Yes. So there's a little bit of discussion about whether a late endosome gets promoted to be a lysosome or whether the lysosome is like totally special. And so was it like not that acidic and then became acidic, or was it always acidic? They're active when it's acidic. So those two pieces are pretty straightforward for Class II. Yep.
So, does this mean that the lysosome, for whatever reason, was gross? That the cell, wouldn't be affected at all by all these proteins that degrade cellular material, or would it just be less affected?
It would be less affected. All right. So on the next figure, I'm going to show you a somewhat adapted view of the MHC Class II presentation pathway. And it's a version from your textbook. And we're going to have to think a little bit about where the sort of problem lies in this figure. So when we looked at the Class I pathway on Friday, we saw delivery as a problem. What does delivery mean? Delivering what to who? Delivering the Amazon package to my apartment? What's going on with delivery? Yeah.
Delivery was getting the piece of the protein, getting the peptide to the same place as the MHC.
Yeah. When we were talking about delivery with Class I, we were talking about getting peptide to MHC. Now let's look at the MHC Class II presentation pathway in sort of vague detail. So if you look at this, MHC Class II, which is is going to be a transmembrane protein. It gets biosynthesized in the ER, as all transmembrane proteins do. So it's in the biosynthetic secretory pathway. The peptides come in, also in the biosynthetic secretory pathway, and they meet up and live happily ever after. Yeah, Mark? Well, so there might be a question about deactivating proteases. But what you can see is that the problem of delivering the MHC or the peptides to the MHC isn't a huge problem. But there actually is a separate delivery problem in MHC Class II processing and presentation. Apparently, even though I totally thought I did not make an edited version of this figure, and instead I'm just going to tell you what I hate about it. So this figure shows you Class I and Class II presentation and processing in the cell. This figure has this dotted line down the middle. In reality, there is no dotted line down the middle. Every cell that does Class II presentation can also do Class I presentation. Every cell that does Class II presentation has a Class II and a Class I in its ER. Every cell that's doing Class II presentation has its Class II chilling in the ER where we're shooting a whole bunch of peptides. So what's the problem here? How do they know to go to Class I or Class II? Or another way to say that is this shows Class II gets some antigen in the endocytic pathway, right? So it shows like Class II is empty all this way. How the heck does Class II not pick up Class I's peptides? It does, but that doesn't mean that the peptides that are all coming through TAP are perfect, size-wise. Some of them are going to be longer. Some of them would totally work. And so the problem here is, how do you deliver a Class II ready to go to these peptides? How is it that this Class II isn't already full when it gets here?
Why didn't you deliver the alpha and beta chain separately?
Well, we're going to see a different answer of how we do this. There is a way we do this, and we're going to see how you do it. And so here, delivery is going to be a little bit of an issue, but it's a slightly different delivery issue. Here, it's how do you deliver the MHC to the peptides, instead of before it was how do you deliver the peptides to the MHC. And there is another protein that works along with MHC Class I or Class II in order to solve this problem. It turns out that this other protein ends up solving two problems for Class II. So it's very helpful for Class II. This protein is known as the invariant chain, or II. The invariant chain has a portion that binds to the peptide binding cleft. And so it blocks the peptide binding cleft, so the MHC Class I peptides can't go there. There's something in the way, which is the invariant chain. You can also see that the invariant chain has a transmembrane domain and a cytoplasmic tail. And as a result, the invariant chain helps MHC Class II get delivered to the right compartment. So basically, we've got something blocking the peptide binding cleft, and we have this sort of cytosolic domain that's almost like a label that says, take this thing to the endosome. And so we have this sorting sequence that allows this to be trafficked to the right place, and also that blocks the cleft. In reality, it actually looks like this. So 3 invariant chains come together, holding 3 different Class II molecules together. And this allows that MHC Class II to actually get directed exactly where it needs to go in this process. So it's both a sorting signal and it solves this purpose of blocking the cleft. See, this is a way better answer, isn't it?
Yeah.
Okay. As the MHC Class II is moving through this pathway. It's going from kind of normal pH compartments to a little bit acidic, to a little bit more acidic, to a little bit more acidic compartments. And as that process happens, the invariant chain gets digested as the acidity increases. So at the end, only one piece of the invariant chain is left. And that one piece of the invariant chain is blocking the peptide binding cleft. That little piece is called CLIP, or the Class II invariant peptide. And so we can see we've got Class II here. It's ready to meet up with peptides. It's got CLIP. It's in the right spot. Invariant chain brought it to the right spot. Now it's here. It's ready to meet up with peptides. So we've got a problem. What is the problem? So we've done delivery. Delivery was done by the invariant chain. Yeah, what's the problem?
How do you unclip the clip?
Now you got to get rid of CLIP. Now CLIP's in the way. And so the place where things get really interesting is binding. How do you get binding to happen? Because CLIP's in the way. Now I want you to think about something for a second. I told you before that Class II, when it has a peptide in its peptide binding cleft, is extremely stable. It's SDS stable. So this is not a trivial problem. You've got an incredibly stable molecule here, and you have to somehow get rid of this one subunit. There is another partner protein that is involved in this process. And in order to explain how this process works, I'm going to do a demo. So, Robert, come on up. Robert's part of my demo. He doesn't actually know what's going to happen, but it's going to be great. Okay, Robert is MHC Class II. He has CLIP. He's holding CLIP, okay? Now, when a thing happens, it will be obvious. Drop CLIP, okay? All right? Okay, so Robert is Class II with CLIP. I am the partner protein. This partner protein in the case of humans is called HLA-DM. In mice, it's H2M. And this is honestly what it does in order to deal with this process. And so that is how CLIP is dislodged from MHC Class II. Excellent job, Robert. So, HLA-DM interacts with the MHC Class I peptide, actually leads to a mechanical force that dislodges the CLIP peptide, thus opening the Class II binding cleft and allowing some type of antigenic peptide to bind. DM will then actually interact with Class II again, and this process will keep going until there is a peptide that is bound with enough affinity that it cannot be knocked out. Yeah?
So,
Yes.
how do you have like the interaction that forces this constant mechanical
We'll talk about that later. But in the moral of the story is we've got our MHC plus peptide, got our MHC plus CLIP, DM comes along, dislodges CLIP. We get a peptide that is in the peptide binding cleft. And so that's kind of the fun step of Class II, which is the binding step, HLA-DM or H2M, that leads to this happening. Immunologists have spent a whole lot of time trying to figure out which compartment this happens in. Is this in the late endosome? Is this in the lysosome? Where is it? What's going on? They spent all sorts of time trying to figure it out. Also, realize that no one ever forgets HLA-DM because it has the demo. So people tried to study where this process happened in all sorts of different ways. And sometimes they're like, oh, it's in the lysosome, or it's in the phagosome, or they go through all sorts of different ideas of where this can happen. And eventually, upon as much study as anybody could come up with, I mean, it's probably a multivesicular body, they decided that the place where this happens is called the M2C, or the MHC Class II compartment. Basically, they gave up and couldn't figure it out, and all of the experiments gave them conflicting data, so they made up their own compartment. And so this process officially happens in the M2C, or the MHC Class II compartment. Although if you look at that compartment, you can see it kind of looks like a Swiss roll. It probably is a multivesicular body in a lot of ways. Then the last part of this process is display. There has been a fair amount of work done on display. And what has been shown to happen is that MHC Class II is actually brought to the cell surface actively by cytoskeletal components, specifically microtubules. And so our Class II will be in a little compartment, like you might see here, and that compartment will actually be brought along microtubules and put on the surface of the cell. You can see that process happening here as well. So here we've got a cell that's going to present antigen. The antigen and the Class II are actually in green. Here's the T cell. So you can see that first the cell starts to make contact with the T cell, and then in fact the cell extends out a cytoskeletal component to interact with that T cell to actually put Class II on the surface to interact with the T cell. Here you can see another example of sort of cells at rest, and then the Class II in cells that have been stimulated, where we've actually pushed all of it to the plasma membrane. And so the person who discovered this, who did this paper, Marianne Bose, if you read this paper, she calls this process tubulation. I'm sorry, Marianne, tubulation is not a word. But there is this microtubule-dependent transport. And so the cell can decide when it wants to put Class II on the surface. This isn't related to the peptide binding in the cleft. And the reason why I point that out is that when someone actually made a mouse that was deficient in H2M, so that mouse had no H2M, it still can have Class II on the surface of the cell. It's just that all of the Class II on the surface of the cell has CLIP in it. None of it has any other peptides, no antigen, just all CLIP on the surface of the cell. So this isn't, it's not like like with Class I where it's sort of held back until it has some antigenic peptide. It can go to the surface when it may be directed to do so. So once Class II displays antigen on the surface of its cell, that will lead to the activation of a CD4 T cell because MHC Class II binds to TCR along with CD4. There are lots of types of CD4 T cells, but the one thing that I can say about CD4 T cells is they all make cytokines to do a thing. So they all make cytokines to influence some other cell type. And so now that B cell might be able to make antibodies better, or that macrophage might be able to kill pathogens better, or something like that. And so here is sort of the overall view of both the Class I and Class II processes. And so at different times I've shown you different parts of this, but this is actually the whole figure. In last time's lecture, when I talked about Class I also told you a bit about how viruses can evade Class I. So you might ask about evasion of Class II. There are 3 big ways that microbes evade class II. I'm going to start with the one that's labeled as number 3. Sometimes they can actually have proteins that modify the phagolysosome to perhaps make it not acidic or to deactivate some of the enzymes, and they can make the phagolysosome a nice happy home for themselves. They can also make sure that their endosome does not fuse with the lysosome, again making that endosome a nice happy home for itself. The other one that I'm going to tell you a little bit more about is an example where, these 2 are examples of things that like Salmonella and Mycobacterium tuberculosis do. We also see examples where the microbe is actually able to escape the phagosome. This is pretty famously done by a bacteria called Listeria, Listeria monocytogenes. Causes a lot of GI symptoms. It can be in dairy products. It can be in all sorts of products. Listeria monocytogenes, when it is in a phagosome, so here's Listeria, here's the phagosome, makes a protein called LLO, or Listeriolysin. Listeriolysin makes a pore and allows Listeria to just break out of the phagosome. So it's basically it's making a pore and then escaping into the cell. And you can see this process happening here in some more detail. Listeria is pretty cool. So we can get phagocytosed into a phagosome. Here you can see it getting phagocytosed. Here you can see it in the phagosome. Then it can decide it doesn't like the phagosome very much. And so it can make some holes in it and escape. Here it is having escaped. It then can actually polymerize actin behind it and make what's known as a comet tail, and actually make a tail that allows it to push from one cell directly into the next cell, so it never has to go outside again. And so you can see it polymerizing actin and pushing into the next cell, and there you can see it with its comet tail in an electron micrograph. You can further see it invading another cell with the comet tail here, and then it will get in a vesicle, break its way out, and live happily ever after. So Listeria is pretty cool. Yeah, Tom?
Is there any sort of specificity for this, or do you think that tries to phagosynthesis?
What do you mean?
Like the Listeria, let's just do it in any cell that it tries to phagocytose?
Listeria is able to use LLO in most any cell that it has. I don't think there's a lot of specificity in terms of who phagocytoses it, but I don't recall off the top of my head. So if you look at this scheme, it's a pretty good way to not be recognized on class 2, right? You see any flaws with this plan? Yeah.
LLO could probably be recognized by an MHC Class I if it gets sent there after being in the cytosol.
Okay. Just LLO?
Or like the Listeria itself.
Or like every other protein in Listeria could also be looked at on class 1. It turns out Listeria is actually beautifully recognized on class 1 and beautifully presented on class 1. So it is a nice evasion strategy for class 2. Doesn't do much for class 1. And so in the end, it just gets dealt with slightly differently. So sort of a meh evasion strategy overall. So one other thing I need to tell you about. Yay. So when we talked about antibodies, and we talked about antigens that could be bound by those antibodies, what did we talk about in terms of biomolecules that could be antigens in the case of antibodies? Yes? Huh? Any. So let's make a list. Okay? 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 Okay. So we got carbohydrates, lipids, nucleic acids, proteins. And remember how there was a thing about they could be folded, or there was a whole folded or unfolded thing? All right. What are the antigens for class 1 in T cells? Or class MHC in T cells? Proteins, peptides. So it turns out there is a way for a couple other things to be recognized by T cell responses. I actually had a lengthy argument with my sister about carbohydrate recognition recently. We'll ignore that. The only thing that I actually want to tell you about is that there is a method for lipids to be presented and for lipids to be seen by T cells. And so we do also see lipid presentation. In talking about lipid presentation, I have to remind you a little bit of a structure of a phospholipid. So note that our phospholipid has a head group that is shown at the top. It's hydrophobic. It's hydrophilic. It usually has some type of charge. And we also have two long alkyl chains, two long fatty acid chains that are hydrophobic. So that's what you need to know about lipid chemistry. And our lipids are presented by another protein, and that protein is called CD1. And so CD1 is our lipid presenter, unlike MHC Class I or MHC Class II. You can see that slightly different lipids can be presented by slightly different CD1s. I'm not going to get super into that. If we look at the structure of CD1, we can see some things that look familiar. So this is the structure of CD1. What you can notice is it looks really similar to MHC Class I. It has a heavy chain with 3 domains, and it even binds to beta-2M, just like Class I does. You will often find, you might find on some old exams, questions about a Class I knockout. It looks different than your beta-2M knockout. Why? And it's because a beta-2M knockout has CD1 messed up too. So we've got this very similar structure in terms of having kind of a couple of helices that are making a binding groove. Everything looks a lot the same. In fact, if you try to overlay Class I and CD1, they look really similar. The only place where they look particularly different is a little bit at the top. And the reason for that is because CD1 needs to have a binding cleft for lipids, not a binding cleft for peptides. And so CD1's lipid binding cleft looks a bit like this. It is much deeper than the Class I peptide binding cleft. It actually has a few pockets that will take those long alkyl chains down in and then leave the head group, the charged head group at the top. And so we can see this nice long antigen binding groove. The lipid actually goes way deep into that binding pocket, and we can see the head group at the top. Yep?
So do the different classes of CD1 present heads that are different?
We will get to that soon. However, there's also another way that we can think about CD1, and that is the steps of the CD1 processing pathway, how it gets its lipids, how it sort of moves throughout the cell. And that part, CD1 processing, looks exactly the same as Class II. So CD1 is in the ER. It moves only through the biosynthetic secretory pathway. It picks up lipids in the biosynthetic secretory pathway and then moves to the surface of the cell in order to present those lipids. So structurally, CD1 looks a lot like Class I, but cell biology-wise, CD1 looks like Class II. Yeah?
Do we have a theory of one evolving from the other?
They probably both evolved from the same common ancestor, not one from the other, though. So you noticed on a previous slide that there was something about multiple types of CD1. In humans, there are 5 different CD1s, although one of them seems to be inactive. And in mice, there are 2 CD1s, one is inactive. What this means is that we know way more about CD1D than any of the others because we can study it in mice and we can make knockout mice. And so a lot of the details of exactly what is different between all of the versions in humans is not super clear because you can't do the same kinds of genetic studies in humans as you could in mice. The things that we do seem to know are that there seem to be differences in terms of which cells have them. So CD1D seems to kind of be everywhere. The others might be restricted to specific cell types. The other thing that is sort of interesting is that they typically seemed to pick up lipids in different parts of the cell. So you can see CD1A maybe is getting its lipids from the cell surface or an early endosome, CD1C from an intermediate endosome, B from like a late endosome or a lysosome. And so those types of things, like where the lipids come from, which cells are presenting, we think are the differences, although those things are not super clear. I told you that MHC Class I is important for presentation to CD8 positive T cells. I told you that MHC Class II is important for presentation to CD4 positive T cells. And so we can also think about CD1 and its presentation ability. CD1 also presents to a unique kind of T cell. CD1 presents lipid antigens, and those lipid antigens are recognized by cells that are known as NKT cells. So NKT cells, once upon a time, people did flow cytometry, and they were trying to detect NK cells, and they found this marker, and they're like, woohoo, it's on NK cells. We got an NK cell marker. And then they realized it was on some T cells too, and they were confused about what was going on. And so those cells got called NKT cells. And so those T cells that sort of confuse them, it turns out, are the ones that respond to lipid antigen and respond to CD1D. So the definition of an NKT cell is that it's a lipid antigen restricted T cell. And we are moving towards things like T cell receptors, and we can talk more about details of NKT cells and T cell receptors later. Remember when we talked about B cells, that there were a bunch of different B cell types, and I told you some of them were kind of innate-ish. NKT cells are one of the innate-ish kinds of T cells. Yeah? This is absolutely different from an NK cell. Yes. Yes. And so now that we know about MHC, we can actually think a little bit more about T cells and T cell responses. So now we understand how this infected cell or cell that has phagocytosed or endocytosed antigen is going to present that antigen. And we can go back to thinking about how that cell that's going to recognize the T cell is going to work. You can remember that the T cell has a T cell receptor, which has two chains, the alpha chain and the beta chain, that are shown here. We've got variable and constant regions. We've got transmembrane domains. We've got CDRs, all that good stuff. Except this is kind of lying to you. So again, just like with B cells, where I remember how I told you there was a bunch of different kinds of B cells. We usually talk about B2 cells when we say B cells, but there's actually more than one type. Same deal with T cells. There's a bunch of types of T cells. And one of the ways we can sort of divide up T cells are into types called alpha betas and gamma deltas. Alpha beta T cells have a T cell receptor made up of an alpha chain and a beta chain. Gamma delta T cells have a T cell receptor made up of a gamma chain and a delta chain. In both cases, one is a heavy chain, one is a light chain. There's always a variable region, there's always a constant region. It looks relatively similar, but our T cells can be either the alpha beta T cell or the gamma delta. Alpha beta T cells are kind of the standard T cells everybody's heard of. Gamma deltas are one of those weird types, kind of like the B1 B cells we saw before. In fact, gamma delta T cells are the ones that you have really early in development. In terms of evolution, they were probably the first ones that evolved. Alpha betas only take over as you get older. Gamma deltas, again, use the gamma and delta gene instead of the alpha and beta gene. But they have differences in their diversity. They have differences in terms of use of CD4 and CD8. There are some questions about their target antigens. They're not found very much in the blood. That actually means it's really hard for us to know how many of them there are, because maybe there's like so many of them like in the wall of your intestine, but since we didn't cut up the wall of your intestine to look, we don't know how many there are. So like because we think there are tons of them in tissues. And so again, we have this sort of innate like cell. Yeah, Mark.
So positive, negative.
Is going to be something we cover probably Monday. And so gamma delta T cells really do seem to be the innate like T cells that may do things like make some cytokines, lyse stressed cells, regulate stromal cells, things like that. So they may be more involved in sort of tissue repair, tissue responses, things like that. The main reason why immunologists, or why I ever had to learn about gamma delta T cells, is that we are going to see that this makes an interesting trick in VDJ recombination in T cells. And so honestly, for most of my life, the only time I knew that gamma deltas were a thing were on the days where I talked about VDJ and T cells, and then I never spoke of them ever again. And so we are going to be getting into thinking about the T cell receptor and VDJ in the T cell receptor on Friday. And so that's why we will care about knowing about gamma delta, because we're going to see it in the VDJ.